a short introduction.
Okay, I will. I'll make it short because I can talk a lot.
Go ahead.
Okay. Hi, my name is Cindy Papali, and it's a pleasure to be here tonight with Brca Strong and Sophie George. It's an honor actually to meet you through via Zoom. I am a breast cancer survivor. I was diagnosed July 2000, stage 1, multifocal invasive tumor. And I had nodes, no nodes were positive, thank God. And along the journey of the 20 years, I'm an author to 3 books, The Empty Cup Runneth Over, Miami Breast Cancer Experts, and Experts in Pain.
Perfect. Thank you so much for joining, Cindy.
Thank you, Tracy.
Tracy, if you don't mind to go ahead and do a short introduction.
Yeah, sorry, I wasn't prepared for this. I was just texting, so.
It's okay.
My name's Tracy Bradley. I'm actually up in Canada, so curious to see the differences between the Canadian system and the US system. I'm BRCA2 positive. I found out after being diagnosed with synchronous ovarian cancer and uterine cancer in 2017. Have a strong history of cancer in our family. My mom unfortunately just passed away in September from a second bout of breast cancer. My grandma prior to that had breast cancer at the age of 40. She survived it, but unfortunately my mom didn't. So just looking at potential double mastectomy here shortly for preventative measures.
Well, thank you so much for joining. Sorry for your loss. And as you have joined us tonight, make sure you stay tuned. We actually have 2 events coming up with High Heel Diaries in Canada. We're talking about the US healthcare system versus the Canadian healthcare system during treatment and during surgeries. We're going to actually talk, our medical director, Dr. Alexi, is going to come on and have a full-blown conversation so we can learn more about each other and how we, how treatment's different or what advances are here over there. Just, I think it's important to share that, different places in the United States have different treatment systems. And I mean, I can relate to that from going to Halifax when I worked in pharmaceutical companies. You guys have a totally different system. And I think it's important that all of us know the differences. So thank you for joining and sharing with us.
Thank you.
So for those of you that don't know me, I'm Tracy Milgram, the founder of Brca Strong, where we eliminate the financial and emotional burden for thrivers, survivors, and survivors. And we are here, regardless of predisposition, to eliminate the feeling of isolation and help you feel whole again. And for 2021, we are honored to announce that we will be funding women pre-mastectomy care packages, which will include 2 post-mastectomy pillows, a drain robe, and a post-mastectomy garment. So if you or your family member need any of this support, please feel free to reach out to us. Again, you could send us an email, go through our website, and as well as we are doing COVID-19 packages, we want to help really focus on the elimination of isolation and feeling whole. And how we do that is by funding different garments, by doing educational Zoom calls, which we just did tonight with Dr. Sophia George, who I was so honored to connect with through some other physicians at University of Miami, but most important, I'm BRCA2 positive. And I learned this at the age of 21 after undergoing 2 lumpectomies at the age of 18 and 19. And as I've always followed the Brca mutation as well as others, I'm interested to see, what's on the other side of what we don't know. And I feel like I never really had a chance to ever speak with a genetic counselor or a scientist geneticist. I feel like, it's interesting in the cancers that we focus on. So to continue to educate. So thank you so much for coming on and volunteering your time. We're really grateful and look forward to hearing your lecture tonight.
So thank you so much, Tracy. Both Tracy's and Cindy, but Tracy in particular for inviting me to talk to this group of previvors, survivors, and everyone else. So let me share my screen. So specifically, well, actually, I should introduce myself. Yeah, so I am a research associate professor in the Division of Gynecologic Oncology and Cancer Control at Sylvester Comprehensive Cancer Center within the departments of obstetrics and gynecology. And I'm a molecular geneticist by training, but I do a lot of work in the context of basic science and across specifically women's cancers, breast, ovarian, and endometrial cancer. So I got my training at University of Toronto in Toronto. So I did my PhD in molecular genetics, and then I did a postdoctoral fellowship at Princess Maverick Cancer Center with a gynecologic pathologist who focuses only on pathology, and she studied hereditary ovarian cancer. And so I spent 5 years doing that. And then I did a second postdoc at Duke University with a medical oncologist. So the first person was Patricia Shaw, where we studied fallopian tube epithelium and BRCA mutation carriers. And then my second postdoc at Duke, specifically, we were studying breast cancer in high-risk women, particularly, again, BRCA1, BRCA2, and in women with who are at high risk for contralateral breast cancer without a known genetic predisposition. And then I was recruited to UM in 2015, so I've been here since. And so my research program and the things that we're doing in the lab and with my collaborations with clinicians, whom I will talk about, those people who they are in a minute, is that we study hereditary breast and ovarian cancer. And we do this by identifying a few ways women at high risk for developing breast and ovarian cancer in our community. We also study how the fallopian tube epithelial cells become cancerous, especially in women with a BRCA1 and a BRCA2 mutation. And I have a gynecologic oncology fellow who is now looking at PALB2 mutations in that context. And then we also study the tumors that arise in women with a mutation and without a mutation. And one of my, I would say, niche interests is studying women from the Caribbean, because I am also from the Caribbean, and we know that in particular in Miami, well, actually everywhere, but in Miami, we have lots of women from the Caribbean, and we also know about the high burden of breast cancer in that region. So for something that you maybe know already, the hereditary breast and ovarian cancer syndrome, what is that? So cancer syndromes produce higher than normal levels of breast, ovarian, and endometrial cancer in genetically related families. So that's that syndrome. And the hereditary factors may be proven or suspected. So what are the signs of people who have a hereditary breast and ovarian cancer syndrome? Usually in family members who have early onsets of breast or ovarian cancer, aggressive pathologies, usually the triple negative breast cancers or high-grade ER-positive breast cancers, high-grade ovarian, serous ovarian cancer. And I will tell you a little bit more just in one slide about high-grade endometrial cancer, where we think that's a link in the disease. And about 5 to 10% of breast cancers, the women who are diagnosed with breast cancers have a part of the syndrome, and about 20% of ovarian cancer patients. And this is a pedigree of an example of a family that we've studied, where in red you can see, so circles mean female, square means male. And in red here, these are women, actually here sisters who were diagnosed with breast cancer. And then one of the sisters had kids who were both diagnosed with breast cancer. And then you see like the subsequent family members. And so that is a family that we would say has a hereditary breast and ovarian cancer syndrome. One of the, I guess, that's another relative of the dad who has who had ovarian cancer. Okay. So what do we know about these genes, at least the genes that we have an idea that are linked to these diseases, a spectrum of diseases? So in the early 90s, Mary Claire King studied families with breast cancer and identified BRCA1. And shortly after, BRCA2 was found on chromosome 13, so here. and was linked to women with both breast and ovarian cancer. So these genes are responsible for the main repair of DNA in normal cells. And when they are mutated, they can no longer repair the mutated or broken DNA. And so cells either then die or live. And cells that get the capacity to live with broken DNA can undergo uncontrolled growth and become tumors. So there are 5 major DNA repair pathways. So one of them is the one that we will talk more about in the next few slides. But there's a base excision repair pathway, there's a nucleotide excision repair pathway, there's a mismatch repair, which is linked to genes that are mutated and give rise to Lynch syndrome family of tumor types, which is breast, endometrial cancer, ovarian cancer, colon cancer. And then you have the homologous recombination pathway, which the genes involved in that pathway are linked to the hereditary breast and ovarian cancer. And then there is a non-homologous end joining. So there are many ways that genes that are important for repairing broken DNA can be transmitted in the germline and unfortunately can increase women's risk for developing cancers. And so here I put the genes that are known to be linked to increased risk for breast and ovarian cancer in the context of this syndrome. Of course, P53 is part of the Li-Fraumeni spectrum of diseases, of cancer syndromes. So what happens to cells that have a mutation? And why are these particular tumors aggressive? So we talk about genomic instability, and I know some of you listening might, and those who have been diagnosed with some types of different types of cancer, in particular breast and ovarian cancer, would think about when maybe your clinician or scientist says that there's genomic instability. So it can happen in many ways in the genome. And usually it's as a result of a mutation in BRCA1 or BRCA2, PALB2, like those list of genes that I just showed, and P53. And what you can see is that here, this is an example of a woman's DNA who had ovarian cancer, high-grade serous ovarian cancer. And what you can see is that there are lots of, so the red means that there are gains in the chromosome, and blue means deleted. And so there are lots of changes that occur in those tumors, and that contributes to the aggressiveness of the biology of the tumor. And then finally, what does the homologous recombination mean? So here, of course, you see, and of course, I'm speaking to an audience that I know is a bit more versed in this context. So we know that in general, when DNA is broken, again, BRCA1 and BRCA2 will come and repair this. But this is a single strand break, and it's PARP is the protein that fixes that. But in the double strand breaks, which is what the homologous recombination is really poised to repair, a person with a functioning BRCA1 and BRCA2 proteins, they can repair this break. But a person without a functioning BRCA1, BRCA2 protein, or those genes within that pathway cannot repair that break. And it is actually, and that, as I said, may lead to, in general, cell death. But actually, in a person with a BRCA mutated cell, it doesn't lead to cell death, like the cells become tumorigenic. And it is that combination actually of having a PARP inhibitor with chemotherapy that's really potent in the context of BRCA mutation carriers, or people who have these genes mutated in their tumors. So this is just another slide that reiterates what I've said in the context of increased risk for breast and high-grade serous ovarian cancer. So we know that women who inherit BRCA1 and BRCA2 have an exponential increase of developing these cancers by the age of 70. And similarly, although thankfully the rise is not as high for ovarian cancer, if you have a BRCA1 allele mutation, you have a significantly increased risk of developing high-grade serous ovarian cancer compared to BRCA or compared to the general population. And so we say that BRCA1 and 2 are autosomal dominant disorder, meaning that you only need to inherit one copy of the mutated gene. And what else can I say about this? So, and then again, these are just the genes that are also linked to these diseases through the syndrome. And the more research that we do, the more we get to understand the relationship of and the risk factors across the different genes. So recently, last week or 2 weeks ago, this paper was published by a group in the Netherlands. And so the title of the paper is Endometrial Cancer Risk in Women with Germline BRCA1 or BRCA2 Mutations, a multi-center cohort study. And again, it was published about 2 weeks ago in the journal, in the NCI journal, so the National Cancer Institute journal. And what they found was that they looked at about 9,000 women, 5,900 women with a BRCA1 or BRCA2 mutation, and about 8,000, so actually it was like 14,000 people, about 8,000 women without a BRCA1 or BRCA2 mutation. And they looked at when the age and how many of them developed high-grade endometrial cancer, I should say. And so what is in the first box here in red, what they found is that for women with a BRCA1 or BRCA2 mutation, they found that 58 of them developed endometrial cancer. And in the Dutch population, so those people who do not have a mutation, they're expected to get, should have 20 people, 20.5 people.
Can I ask a question?
Yes.
How has endometrial cancer always been attached to the BRCA mutations, or is this like newer research found?
So it's been always loosely attached.
Okay.
And what we did not know is a big cohort study like this, right? Because there was a paper published by Noah Koff, he is now at Duke University, and showed that there was an increased risk for women who had a BRCA1 mutation to develop high-grade endometrial cancer serous type.
Okay.
And there was a paper published a few months ago in England saying that there was no relationship, but it was a small cohort. So of course, the larger the study, the more power it has to be able to identify these types of relationships. A colleague of mine can speak at more depth about the types of mutations that are found in tumors that arise in endometrial cancers and why we think that there is a similarity, or I can talk about it a little bit too. So what they found is that there was, the study provides really strong evidence. Let me see before I summarize. So yeah, so they found that there were more people with a BRCA mutation, 1 or 2, who developed endometrial cancer. And that was statistically significant, as you can see here. And then they looked at different types of endometrial cancer. Just like there are different types of breast cancer, different types of ovarian cancer, there are different types of endometrial cancer. And in particular, there was a really strong relationship between having a BRCA1 or BRCA2 mutation compared to, because this is a very rare, relatively rare tumor type, and the risk of developing endometrial cancer. And so the study provides really solid evidence that BRCA1 and BRCA2 germline pathogenic variants of germline mutation carriers have an increased risk of endometrial cancer by at least 2 to 3-fold. And additionally, they find that the type of endometrial cancer, which is again very high grade, very aggressive, is linked, closely linked, and even in the endometrioid, linked to having a BRCA1 or BRCA2 mutation. So I'm going to actually, with that slide, segue into some of the work that we've done in certain populations, the Afro-Caribbean and African-American women. So because of this publication, we also knew that women, African-American women, are disproportionately diagnosed with this serous kind of endometrial cancer. And so there is a lot more work to be done in the context of identifying these populations within the United States who have different ethnic groups, different race groups, racial identities, to understand the relationship between the predisposition and this particular type of tumor. So a group that I study because of the effect of cancer on that particular population is Black women. So Black women and African-American women are more likely than white women to be diagnosed with breast cancer under the age of 40. And about half of the breast cancer cases in Black women are diagnosed at the local stage, so compared to 64% in white women. So it means that the women are diagnosed at advanced stages, at more than half of them. And of course, why that is the case reflects we know about the socioeconomic status, access to care, but there's also a relationship that we are studying in the context of biology. And so African-American women have the highest death rates of all racial and ethnic groups, and are 42% more likely to die of breast cancer than white women. And that breast cancer is the most common cause of death amongst Hispanic women. And of course, our state is full of Hispanic women. So it's an important aspect of that population to study. So what do we know about where we are? So I'm at the University of Miami, and as you can see, we're surrounded by downtown, well, it's not really downtown, but we're surrounded by many neighborhoods that are with majority Black people, and actually Hispanic too, one so. And in general, about 10% of the Black population in the United States are immigrants. And the immigrants are mostly from the Caribbean compared to Africa. And then Florida has the second largest Caribbean population in the US, the largest is in New York. But we also have a very disproportionately large population of Haitians and Jamaicans in our area. So a few years ago, my colleague and mentor actually at the time made an observation in the clinic. So this is Dr. Judith Hurley. She's a medical oncologist, and she treats women for breast cancer or with breast cancer at Jackson. And one of the things she observed while she was treating these women is that a lot of young women who happen to be Bahamian were being diagnosed with aggressive and advanced stage disease. And so, of course, we know that those are two key points about maybe etiology. So how does, why is it happening? And so we know that early stage onset, early age at onset, and being diagnosed with a high-grade tumor, and of course, family history, linked to hereditary cancers, breast cancers. So what she found, she did a little study, and she looked at the age distribution of the Bahamians who were diagnosed with breast cancer. And she did that with colleagues at Princess Margaret Hospital in the Bahamas. And what she found was that about 53% of the women that were diagnosed with breast cancer were under the age of 50. Now in the US, the average age of onset of diagnosis is of 63 years old, and about 57 in Black women. And that 44% of the women were diagnosed with advanced disease. And so she did a study that was initially funded by Susan G. Komen and the Bremen Foundation for 250 women And what they found was that there was a range between 24 and 28% of Bahamian women who were diagnosed with breast and ovarian cancer had a deleterious mutation in BRCA1 and BRCA2. Now, what do we know? And this was published in two different articles. Tyler Donenberg, who is a genetic counselor at UM, is the first author, and Dr. Hurley is the senior author. And so what do we know, right? So what do we know about hereditary breast and ovarian cancer in the broader aspects of things? So we know that, well, I have it 3 to 5%. It's more like 5 to 10% of breast cancer cases are have a germline mutation, and about 20%. And that in the US, between 1 in 400 to 1 in 500 people have a germline mutation in BRCA1 or BRCA2. But we also know that there's some populations, smaller populations, like the Ashkenazi Jewish population, who has a very high rate of germline BRCA1, BRCA2 mutations in their population. So there was a lot of work done in the Bahamas. We looked at women, so we went back and we looked at women who had a, we did a screen, a population-based screen in about 3,000 people, and identify, well, how common is this mutation in the general population? And we found that about 1 in 1,000 unaffected women without a family history had a germline mutation, so which is like very, very low, okay? But then if you looked at the people with a family history, it was 1 in 35. And that about 38% of unaffected women with a known mutation in the family carried a familial mutation. So it means that if you have a family history of breast cancer and you're Bahamian, there is a likelihood that you yourself may be a carrier, and that's important in the context of prevention. Tracy, is particularly what this, your foundation and your this is about. So we then did a bigger study. We just published this month, actually we're still in March 1st, where we asked the question, well, is this phenomena, high rates of hereditary breast and ovarian cancer across the entire Caribbean region? And so we conducted the study in 7 countries that are listed here: Bahamas, Cayman Island, Dominica, Haiti, Jamaica, and Trinidad and Tobago. And what we found was that Bahamas still, unfortunately, had a high rate of inherited breast and ovarian cancer. We looked at this in 1,000 women. We did panel testing, so we studied beyond BRCA1 and 2. So we looked at 31 genes, and we saw that Barbados had 17% positive rate, and Trinidad and Tobago also had a high rate, higher than expected, but lower than the Bahamas. So what do we know about, so this is now going into the islands where we are focused on small populations, but of course these people in the islands are represented in the United States and Miami, as I said, in particular South Florida. So we also wanted to, so we looked at the data that's available about African-American women, because as I said initially, that a lot of women are being diagnosed young. And so the rates actually of inherited cancers is not exceptionally high. It's within the range of 5 to 10%. So there are different studies that have been published in different parts of the US. And so in Florida, Tuya Pal, who is now in Vanderbilt, before she was at Moffitt, found that 12.4% of the women who are diagnosed with breast cancer have a mutation in BRCA1 or 2. And then in Detroit, they recently, last year, published it's about the same. And then in a national study, the rates were a little bit lower. So of course, like identifying women in different parts of the US. And so I will stop here, but just to summarize, what we found in actually the study that we did in the Caribbean is that there were few recurrent mutations across the region. So like I know some of you may be aware that there is the Ashkenazi-Jewish mutations and some other mutations that are very well known. We didn't find that phenomena across the region. We found that different countries had different types of mutations that were essentially unique to the patient population. And that in some countries like Trinidad, that there were multiple mutations across the entire spectrum of the gene. And so there is no one, for example, one mutation type for certain countries. It's important to identify families at high risk for developing breast and ovarian cancer in our community, because we know that there are ways that we can prevent these diseases. And of course, I'm saying that not in the context of access to care, clearly. That's another kind of talk. Access to testing across the region. So we looked at, we're doing this kind of type of work in the Caribbean, and how is that going to impact the entire infrastructure of the healthcare system? And so we've been working actually with different ministries of health, with different clinicians to implement testing, and they have implemented testing in their clinics so that they can provide this service to their patient population and community. And we are in the process of studying the tumors that arise in these in the women who have these mutations, because we also know that having the mutation itself is not necessarily, it's a likelihood that it will give rise to an aggressive tumor, but we know that in the context of overall survival, and when women are treated with the standard of care therapies, that some women are not responding to the PrEP inhibitors and are not responding well to the systemic chemotherapy. And so we are studying these tumors to try to get at, like, is there a spectrum of mutations that some people would benefit for different types of targeted therapy in our community. And then with Yodea, we are really trying to do community-based testing. I say trying because we're building the program to do that. And of course, with that comes like different opportunities for improvement in our approach. And then finally, I spoke a lot about Black women in English-speaking and the Haitian population. But we got a small grant through Sylvester and the Global Oncology Program to pilot a study in Dominican Republic, because we know, and the reason we're going to the countries is because it's easier to enroll a population of people from that country and then be able to disseminate and apply the information to a broader context. And so we in May, we'll be doing some genetic testing through the research project in the Dominican Republic. So with that, I will stop, right? And acknowledge my collaborators and colleagues, in particular, Judith and Talia, island hopping and doing some work in the region, and Stephen Narod, who has been really important in the context of characterization of the mutations. And these are all of our wonderful collaborators in the region. And then at for this particular work, we work very close with Dr. Carmen Gomez. She's a breast pathologist, and Dr. Matthew Stambrak, he is the interim division chief of gynecologic oncology at Marilyn Wong, who runs a lot of clinical trials, gynecologic cancer clinical trials, and Andrew Pinto, who is a GynPath. And then, of course, my collaborators across the different sites in the US who are also part of the African Caribbean Cancer Consortium, and then cancer societies, and of course, funding. So Susan G. Komen funded the project in the Caribbean. We got some funding from Color Genomics from Color to do some testing. And then I am supported by the Department of Defense for my wet lab stuff that I think about in the lab all the time, and the fallopian tubes, and I'm hearing ovarian cancer whispers as I am a fellow, recently awarded a fellowship from that foundation. So that I will stop and I can answer some questions.
Perfect. Thank you.
I have a question.
Go ahead.
Yes, I'm going to stop sharing my screen.
Hi. Okay, my question is, since I've waited so long to have this genetic testing, my nieces and great nieces are asking, how will they be affected with the results of my tests if certain ones come out positive. I know that the mother and daughter is close, but how is it with my niece and great niece?
So with your nieces, so the nieces, so it's your sisters or your brother's children, right?
Yes, it's my brother's daughter.
Right. So they will, I mean, you inherited if you have a mutation, you inherited it from your mother and father. And so your sisters and brothers have the same chance of that you had in getting it. And their children will have the same 50% chance. So I would say that if you have a mutation and they're of age, you should, and that's a great thing to share the information. That's what we are telling everybody, to get to share it, not keep it secret, because we can save lives. So yeah, so tell them what it is, and then hopefully well, I'm assuming that they're old enough. They're 21. I know Tracy.
Yeah, she's 40, and one is 43, one's 40.
Right. So then they should definitely get tested, and if you have a mutation. I would say if your mom or somebody else has one, they should also get tested. Like somebody, Yes,
my mom was tested and she was negative, except for BRCA2 was inconclusive.
Right. Well, but you also have a strong family history of cancer.
Yes, my mom was diagnosed with cancer of her at 77 of her cervix, and then when she was 87, she got breast cancer. Her sister had it at 46, and my father's sister had it at 36. And to add insult to injury, I'm also a DES baby. That was a drug put out by Eli Lilly in the 50s to prevent miscarriage. So it's been known to cause breast, cervical, and colon cancers. So that is another story with the DES. That's a drug that was to prevent miscarriage, but in the same time, 25 years after, they found out that it can cause other cancers.
Right. So I would say that your nieces should speak to a genetic counselor to assess their risk and to determine what's the best approach for them.
Okay. Yes, I'm going to let them know, but I just had it done yesterday, so it'll come back. I think she said in 2 weeks because they're testing a lot.
Yes.
Oh, I thought it was only 40 tests. She goes, no, we're going to do all 80, but that's Dr. Kalfa.
2 weeks? I had 2 weeks, 6 weeks.
Yeah,
really? The technology is moving actually quite quickly. I did my testing in December in solidarity with a family member, and because she had a strong family history and I don't, but we're related. So I was like, okay, I'll do it with you. And it only took 2 weeks. I was surprised. Actually, I was expecting it to be longer. So.
Well, I mean, I was in 2004. That's when I had them. So I mean, like, think about how far everything has come.
Right.
We can sequence a human genome like in 5 weeks. You get the results. So yeah, the whole genome. So yeah. Any other questions?
Well, those stats sure were an eye-awakening for me. And if there's anything that we can do at Brca to help promote, bring women to you to get testing, like obviously, as you know, we're already working together, like what we can do to promote to get women enrolled and how we can continue to make a difference, whether it's genetic or whether it's breast cancer, ovarian cancer, endometrial, like just you coming on tonight, I learned that endometrial cancer was, included in BRCA. And to be quite honest, I have never heard that. Hey, Dr. Alexia, I was like, what do you mean? Like, this is something so new, but this is the purpose of why we do what we do, is to continue education and making sure that we know what's happening. And obviously over the years, right, things are evolving, things are changing. So it was just very interesting to me. I know we had a couple of people join late, but like the stats and stuff, I can't wait to share it, to let our community know, like, these are the stats, ladies, like, step up. Like, we've got to make a difference. I mean, we already know the higher mortality rate in African-American Black women, right? But like, let's now get them to enroll in clinical trials. Let's control to get that data that we need, because yes, white women, Caucasian women could keep enrolling, we could keep going, but we need stats on African-American Black women and Caribbean and Dominican Republic, like you said. Like, I'm absolutely like, I'm shocked.
Yeah, and so it's, as you said, it's really, so it's two things. One of the things that we've been told, so we've done work, Dr. Schomburg and I in Opelika, where we're just like having, low key, not low key, but just like regular conversations with women so they can ask us questions. Because we realize that there's two ways. One, we have to open the doors and say and recruit actively for participation, but also explain what it means to participate in research. And that's maybe another talk another time. Because of the history of being abused or unethical activities in the context of biomedical research. But it is so important for us to participate because that's how we're going to know more about what's going on. And that's in the broader context. So health inequities, socioeconomic status, whether it's biology or not, like we have to now all like try to put all of it together to get a better picture of what's happening so we can improve the outcomes of some groups of our community that all of us are important, right? We all want to hang out and be around for family members and friends, and especially as women, because we're so involved with our broader community, school, everything. And so it's really hard to see young people in the prime of their lives being diagnosed with diseases that some of them are preventable, in the context of also knowing, having the information, right? And I can like, yes, having the information and not being able to do something about it is another aspect of but we still at least now need to start talking about it. So if people have the information, they can act on it. If they don't have it, then we can't do anything about it.
Dr. George, what is the stats on Asian women? I didn't see any stats on Asian women.
That's a good question. I can say, I'll just be honest and
Because
say,
people listening might want to know. Yeah,
I do not remember because I just, that's a part of the population that I have not actively studied. In the Caribbean, we have a small population of Asian women, and some of them did participate in our studies, and we found one common mutation, but I can't speak about the breadth of genetic predisposition in Asian, meaning Indian or Chinese, or Japanese population. Although that information is out there, it's just not actively spoken about, I guess, because maybe in the US, we're not actively studying that. Or maybe someone is and I'm not aware.
Or maybe the risk is lower for them for some reason, I don't know.
So that is true. In general, when we look at the epidemiology of breast cancer, the risk of triple negative breast cancer is lower in the Asian community, but in the Native American community, it is high. Like the breast cancer mortality is high. In the Latin American community, Hispanic, I'm painting a broad brush here in terms of that particular population because they are not a monolith either. It's also high. And who else can I think of? Yeah.
Does anybody else have any other questions?
I have a question.
Go ahead.
Hey,
Dr. George. Great to see you. So Dr. George, this is Dr. Gaffney. She's actually our medical director as well. So
hi. Nice to meet you. I was only on for the latter half of the talk, so I apologize for arriving late. But super excited to see the work that you're doing with the University of Miami. I'm actually applying for a job there. So
are you coming here?
I don't know. I'm applying for a job though, but I didn't get on to say that. I got on to ask about the genetic outcomes of the Caribbean women, right? You said that there wasn't a continuous or a specific mutation that pops up like the Ashkenazi variant for the Brca gene mutations. Is that information similar to, comparable to, in any way related to when we here are told, and I guess I'm asking specifically for Black women, would those particular mutations come up as variants of undetermined clinical significance if they were looked at in an American cohort, or if they were studied against the currently available human genome? Because my understanding is that Black Black people are more likely to test positive for variants and be told that they are of undetermined significance. But is that the same as, or is that different than these variable mutations that you're seeing across the Caribbean population?
Right. So let me answer that in parts. So in the Bahamas, we found 5 founder mutations in BRCA1 and 2 in BRCA2. So those mutations are recurrent in Bahamas, and they're pathogenic. And in Trinidad is an island that has a big mix of people. So they have Indians from the ancestral Indians, Southeast Asia Indians from the Indian continents, Chinese as a result of the waves of people who came post-slavery, post-abolition of slavery. And so we saw mutations that have been identified in those particular populations present in Trinidad population. So now in the context of the variants of unknown significance, that's what you said is correct. Because we have been under tested, under participated, there's a lot of results that are coming now that we're doing tests and finding that many of the tests are coming out as a VUS, so variants of unknown significance. And we found about 50, I think, of those in the Caribbean too. So it's the same thing. We hope that, and that's part about doing science and collaborating. We hope that as me and my group and my collaborators are including more and more women across I would say the ancestral spectrum. So white women, Hispanic women who are white, Black, Native Indian, Caribbean women with all the mixes, that as we do more and we look at the trajectory of their tumors for those people with cancer and look within the tumors, we can have better ideas of whether this is a VUS or whether it actually is a pathogenic variant. Because the only way to know is to actually do the research. And that is, some people are doing that. Maybe not in the Black population, actually, but it is under study in the greater aspects of what's happening, because more and more testing is being done. The VUSs are being identified, but disproportionately, unfortunately, in African American and in Black people because of the deficit of data. I hope I answered the question.
You did. Thank you so much.
Yeah.
You did. Thank you.
Does anybody else have any questions?
And welcome to Miami, Lily. Sending it out there.
Well, thank you so much for coming on and sharing all this information, and we look forward to having you back on in the future. And for those of you that are listening, make sure you share with your family and friends, especially in the different ethnicities in the world that we live in. This video could change a family's life. Thank you again for coming on, for your dedication and time. We're super grateful. I hope everybody has a great evening.
Thank you so much.